And the first lecture is important. You'll get to see my lecture style. I may differ from other instructors. I'm not as tightly knit as some other instructors. I kind of like more have a free flow of conscious, and whatever I think of that's important, I write it on the board. And it's important that you take good board notes and let that be your study guide for the class. Okay? So the first chapter is endocrine, and the first illustration I'll present to you is this. This is kind of the illustration of the physiology. Let me define the endocrine system. The endocrine system consists of endocrine glands, or a cell, which is pictured. Endocrine glands synthesize chemical messengers called hormones. Which are secreted into the bloodstream. Hormones travel to specific target organs. Hormones travel, well, they travel in the bloodstream as pictured. They travel to specific target organs. The hormone elicits some response from the target organ. So I'd say the key words there for studying physiology, you got to study the glands themselves. We'll go through them one by one starting today. You got to know the hormones that they synthesize, secondly. And well, you got to understand that the target organ is going to influence, and of course the physiology part is the response. And so they call these reflexes, these endocrine reflexes. And this is the process. The gland secretes a hormone. It affects something far away because blood goes everywhere in the body. There's some specificity. Hormones don't affect everything. They only affect a specific target cell. So there is specificity. So they're circulating, it finds its target, you get the physiology. And so this first hour of lecture is just basically going through examples and the generalities of the concepts of how hormones work. So the first concept is they're long distance. They use the bloodstream, and blood goes everywhere. Now you talked about chemical signals before. Usually we teach neuro last in 430. Remember studying the synapse, the neuromuscular junction, that's an example. Neurons, action potential, communicating with other neurons. The synapse is a very small space. Those neurotransmitters travel a very short distance. Hormones use the bloodstream. They travel very long distances. But both neuro and endocrine, those are the two major systems in human physiology for communication. Okay, so that's why we spend a lot of time on them. As an example, I like to start with this one. This is the regulation of blood sugar. Example, regulation of blood sugar, blood glucose. Glucose, you need to know, it's the circulation form of sugar in the blood. So, the reason why we need this communication system, something goes out of whack. Okay, like something gets too high or too low. By design, like for example, you eat pasta. Your blood sugar will go up, increased blood glucose. And you want to take that nutrition absorbed from your intestinal capillaries. You want to get it out of the blood into cells for use. And so you need a detection mechanism to correct this imbalance. Fasting blood glucose should be less than 100 mg per deciliter. Okay, so when blood sugar gets high, because you ate a meal, okay, the gland that's stimulated that can detect this imbalance is the pancreas. So here's kind of what the pancreas looks like in inverted tobacco pipe, pancreas. Behind the stomach, they just kind of have it floating out there in space on the figure. But anyways, it can detect an elevated level of glucose in the blood. So in response to that, it'll secrete insulin. Maybe insulin, I symbolize that with these green balls they enter the bloodstream. Let's preface this by saying, when you ate blood glucose, let's say glucose was already in the bloodstream. Let's say glucose is the blue balls. So let's put the blue balls in there. Let me extend this blood vessel tube out to here. And the glucose entered the bloodstream. And the pancreas had an ability to detect that increase in glucose. And in response, insulin was secreted. So now what you have is insulin and glucose circulating together in the bloodstream. And to correct the imbalance, you need to pull the glucose out of the bloodstream. The insulin can do it. So what does it do? It goes to its target tissues. Maybe it goes to the liver, maybe it goes to the skeletal muscle. Both of those organs can utilize the glucose, so they will present the insulin receptor. Muscle, skeletal muscle, and liver. Liver, biggest solid organ in the body. It's in the abdominal cavity. For now, just know that liver, skeletal muscle, they both have cells that will present a receptor for insulin. So let me draw a generic looking cell here. Could be from either organ. And it'll present insulin receptor. And then so when insulin is secreted in appreciable amounts, it'll diffuse out of the blood vessel and it'll bind to its specific receptor. And that will trigger a response for this cell to take glucose up. Okay, so maybe glucose is coming secondarily. Maybe there's some kind of glucose vesicle in the cell that can kind of take up the glucose. Right, and that way the glucose can enter the cell. What do cells do with this fuel? Well, they can burn it. They can make ATP out of it. You can study glycolysis. They can store it for energy. For example, glycogen is a chain of monosaccharides, simple sugars. So if you want to store the sugar you eat, glycogen is a polymer, glycogen. It'll take the monomer and it'll add it to the chain to increase your glycogen stores. Okay, that's why on the figure it says that. The conversion of glucose to glycogen, right? You're increasing your glycogen stores by storing it in the skeletal muscle and in the liver. So the net result is, by pulling glucose out of the blood and into the tissues, the liver and the skeletal muscle, you do the opposite of this. Decrease blood glucose. Let me ask you this. What if this happens, insulin binds, but then you don't take up the glucose? What's the result for the blood glucose if it can't accomplish this glucose uptake? The level remains high. That's diabetes, right? It's a problem with insulin. Okay, we can get into it later, but when it works, when the insulin-sensitive tissues work, you should lower the blood glucose. Your body is 80% muscle. If there's a sign of insulin resistance, it's going to be from the muscle. But normally this is how it works. So, take inventory of what we did here on my little diagram. I'll just kind of write it over here on the side. Number one, we noted the gland pancreas, check, went through that. Okay. We studied the hormone that it secreted. Insulin, okay, check. And thirdly, target organ. Well, there's a couple. We listed skeletal muscle and liver. Okay, check that box. And I don't have it as a number 4, but we did note the physiological response, or just response. All this happens to reduce the blood glucose. Okay, that's ultimately the response. And that's kind of how you do it, as a generic example. Let me get some real quick. Sometimes, just to break the monotony of lecture, I like to ask what I call half-sheet questions, and this is actually worth credit. Whenever I do that for a given day, just write whatever half-sheet questions I ask you to do on here, and I collect it at the end of the day. That's how I monitor attendance, and you'll get a couple of points. You don't necessarily have to write the correct answer, but you should be within the ballpark, right? Because it's like we're trying to learn here. So I'll pass these back. And put the leftovers over by the door. What I want you to do on your half sheet, I'll wait for you guys to pass them back. Yeah.
So you won't have insulin in the cell unless there's high blood glucose.
That's right. It's only released in response to high glucose. In fact, if you have high insulin levels, I know I'm erasing it, but that means insulin's not working. They call that hyperinsulinemia, and that's an indication of diabetes. So what I wanted you to do on your half sheet, see this 1, 2, 3, 4? Do that on your half sheet, but do it for the other imbalance. Do it for the imbalance where the stimulus in is not a rise in blood glucose, but a drop. Okay, go through the same process and just outline 1, 2, 3, 4. Keep it short. Okay, because those sheets of paper aren't too long. Decrease blood glucose. So I'll give you a couple of minutes to do that. This isn't a quiz. You can talk about it. I encourage conversation during this time. It's a little break for me. It's an opportunity for you to think. I think that was enough time. I'm going to move on. I'm interested to see what you put for the bottom part of the figure. But feel free to restate your answer if you want. I won't collect the half sheets until the end of lecture today. But moving on to the next slide, it's a couple of terms. We're talking about the endocrine system, but I at least want you to know what the term exocrine means and how it's different. They're both glands, but the function is different. You know, an exocrine gland is a gland that will secrete pretty much everything that's not a hormone. That would include things like saliva, tears, sweat, enzymes, non-hormones. And they use a duct so they can secrete it to the surface. It's basically secreted outside the body onto the skin surface or into the digestive tract. Okay, hence the term exo means outside of. So they use a duct. These glands have a duct. Secretions are on skin surface or in GI tract. Inside the GI tract is considered outside the body because it may pass right through you. All right, so anyways, they're derived from epithelial cells. That's the tissue type. I usually abbreviate epithelial tissue, ET for short. You get used to that. Then contrast that with endocrine. That's our hormones. And they don't have a duct. They're ductless. But they do use the capillaries. They use the bloodstream. The capillaries are the smallest of the blood vessels that can take up the hormones and get it in the bloodstream. Now, they're derived from epithelial tissue as well. I'm writing it out this time. But there's also hormones that can be secreted from neurons. That's something you may not have heard of before. Neurons secrete hormones. I'll write both of them on there. So let's just kind of compare and contrast. The picture on the next slide kind of shows you that difference between the ducts and the no ducts. The exocrine slide, you can see the duct remains as a conduit. So the secretory cells on the bottom here have that function. And the cells forming the neck here, they don't have a secretory function, they just act as a duct. So you have a straight shot to the surface. There's no duct here. It disintegrates in development. And so these epithelial cells, it's a cluster of cells that will secrete right into the bloodstream. Okay. Now all endocrine glands are illustrated here, and we'll start to go through them. For your quiz on Friday, what I'll have to do on Wednesday is I'll start teaching the histology of all of these so that you can be prepared for your quiz on Friday. My strategy for that is there's a scope right there, and you can see there's a camera on top. The camera is hooked up to our PowerPoint slide. As I present the slides, you'll be able to see what I see. And I can even take pictures with the camera and give them to you on Canvas. I use those for the quiz. Okay, so you're studying the unlabeled pictures of the histology slides. So it's important that you pay attention when I'm presenting it. It's pretty straightforward. So in that way, these pictures will make more sense. They may not make sense now. But right now, I just want to stick to the basic general physiology before we get to the histology. So when we talk about hormones, we like to mention factors that affect the target cell specificity. There's a lot of detail that go on to make sure that these can fire a response. So let me erase the board again. Factors affecting target cell specificity. Well, number one, I got these fluid compartments here. Appreciate that a hormone secreted into the bloodstream has to diffuse out of this fluid compartment, the blood vessel. Okay. The plasma is the liquid part of blood, and hormones are in there. It's got to diffuse out of the blood vessel through the interstitium, which is the second fluid compartment. That's a trigger response in this cell. Okay, the fluids inside the cell are considered a third fluid compartment. So one factor is you have to secrete enough hormone versus not enough. So what's enough? Let's call it hormone concentration. Here's my chemistry brackets, my concentration brackets from chemistry. If you secrete, well, they're very sensitive, so you don't have to secrete too much. Usually books will say concentration 10 to the negative 9 to 10 to the negative 12 molarity, okay, is enough secreted. If not enough is secreted, you won't trigger a response. So that's one factor. Another factor is this up or down regulation of receptors. So let's look at the figure. If the target cell kind of senses rising levels of the hormone, it'll prime itself by upregulating the receptor for that hormone. If the level of the hormone stays too high, you don't want to overshoot homeostasis, it'll downregulate the receptor, so you don't overshoot homeostasis. So, downregulation, target cells lose receptors in response to the hormone, in response to persistently high hormone levels. That'll be downregulation. So that's a factor. Is the receptor there? Another factor is affinity. This is desirable. There's a nice tight fit. Because that'll trigger the intracellular response. I mean, this won't. I mean, note the difference between the hormone and the hormone receptor. They have to fit tightly. Okay? So the illustration, you know, if it's low affinity, you won't trigger a response. So now we can kind of start to talk about the different varieties of hormones. Well, there's three categories. I kind of boil it down to two: proteins and steroids. Proteins, the monomers are amino acids. An amino acid has this basic structure. It's a carbon surrounded by 4 positions. A generic R group, that kind of changes depending on the function of the hormone. A hydrogen, a carboxylic acid, and an amine group. So that's the general structure. That's the monomer. That's a single amino acid. But link those together with in a polypeptide chain, they form different structures. And I think the thing I would note, protein hormones are water soluble. They're not lipid soluble. Well, what that really means for the cell is, here's the target cell, there's the cell membrane, there's the nucleus in the middle. If it's going to be responsive to a protein hormone, it must present the receptor for that hormone. Let's say this is the hormone. The receptor must be presented at the cell surface because the hormone cannot diffuse past the lipid bilayer of the cell membrane. Okay, so in that way, the high affinity relation at the surface will trigger an intracellular response by binding on the outside. And they call these intracellular molecular biology pathways second messenger systems. Because of the nature of the protein hormone being not lipid soluble. You bind at the surface, basically. Now, steroids, they have a different structure altogether. Steroid hormones are classified as lipids. And they have that rigid ring structure. And they're all derived from cholesterol. Well, that's cholesterol, but there's a couple of examples of cholesterol on the top and then the sex hormones right underneath it. You can easily convert cholesterol into any number of steroid hormones. The sex hormones are just an example. Because steroids are classified as a lipid, these are lipid soluble, not water soluble. And because of that, they can bind their receptor inside the cell. So say, for example, here's the cell membrane, here's the nucleus. You can have a receptor that's in the cytoplasm or in the nucleus, and then the hormone can bind in either location. So it doesn't have to bind at the surface. And it can, if it does, it can like directly turn genes on, and so they kind of call this mechanism where you kind of bind directly, whether here or here, to DNA, to genes. They call that direct gene activation. So those are the kind of two mechanisms for the proteins or the steroids. There's a third class. Some consider eicosanoids hormones. I do not. Don't worry about them. So the good news for the day, they're not responsible for eicosanoids. So I put the slide in there to remind me to tell you that. Okay? Not responsible for eicosanoids. But let's talk about generically how proteins, steroids, how they are triggers, okay? They're secreted, great, but if they don't trigger a response, you're gonna keep secreting the hormone until you get the response. So here's the trigger. I wrote direct gene activation first. I wrote it second on the board there. But you go straight shot to the genes, turn them on, get the response. Because you kind of have to like knock on the door, so to speak, you have to act through second messenger molecules, and the G proteins are the commonest ones I usually present. A G protein is an example of a second messenger system. There are others. Here's the example of, well, it's the professional picture of direct gene activation. So, see if you're with me. What color is the hormone? Yellow. What color is its receptor? Orange. And they want to show you, when you bind, you form this receptor hormone complex, in this example, in the cytoplasm. Number one, form receptor hormone complex in cytoplasm. Step 2, you enter the nucleus. Step 3, you bind DNA, you turn the genes on. So let's go back. Step 2, enter nuke. Step 3, bind DNA, response elements. Fancy way of saying turn gene on. You're going to make the proteins that can get you your response. And so that would be step 4. You do the transcription. Step 5, translation. Step 4, transcription. You're transcribing the gene. Remember, that's mRNA. That has the code on how to make the proteins. You just transcribe the code, kind of decode that translation in the cytoplasm. That's where you get your polypeptide protein. I'll say new protein. Okay, you synthesize it. We teach the details of transcription, translation in 430. But that's what you got. You're turning on genes. That's what the figure is trying to show you. Same thing. You're triggering a response in a different way for the protein hormones. It's over here. Okay, number 1. Okay, look for the little pink balls. It's on the left. That's where this figure begins. That's the hormone. Now, it can't enter the cell because it's not water soluble. It's not lipid soluble. It can't get past the hydrophobic zone. So it's got to bind on the outside. Okay, so that's our hormone receptor. The hormone is considered the first messenger in this second messenger system. So the hormone binds its receptor on cell surface. Okay, and what that will do is that will trigger and activate the G protein to move along the inside of the cell membrane. This right here. It's activated with this phosphate called GDP. Let's just say that the activated G protein it moves, okay? In step 3, activated G protein moves, so in step 3, what happens is adenylate cyclase will be activated by that activated G protein. And that's the molecule that will actually generate your cyclic AMP. So all right, G protein activates adenylate cyclase. Step 4, adenylate cyclase it converts GDP, to cyclic AMP. Okay, well actually no, that's not GDP. It converts, let me write it out, it converts ATP. Think of ATP as 3 terminal phosphates. You cleave off the last 2 to leave you with 1 P, mono, and we just call it cyclic AMP, or CAMP, C-AMP. That molecule, C-AMP, is considered your second messenger. There are others. For example, calcium is a popular second messenger in a lot of systems. So I'll put that in parentheses because that's not this one. Calcium ++. But for this example, it's cyclic AMP. Now, if you look at the figure, I'm going to stop there, but they kind of like talk about how that will activate kinase activity, and you'll get some kind of response. And they give the example of a relay race, passing off the baton, right? Receptor, G protein, enzyme, second messenger, cyclic AMP. Okay, I like to use the analogy of you go for a walk in the nighttime. Say it's me, I'm walking around my neighborhood, and I see my neighbor's house is on fire. So I pound on the door, and I can only manage to wake up a small child who's trained not to open the door to strangers. So I'm like, hey, let me in. There's a fire. I'll help you put the fire out. And the kid, no, I'm not gonna do it. So I'm like, okay, go wake up your parents. Your parents wake up, and they call 911 or put out the fire, get the fire extinguisher, whatever. The point is, I never enter the house. Okay. So in my analogy, what am I? The first messenger, the hormone. And I never enter the cell. I never enter the house in my analogy. So the small child is kind of like, this activated G protein that moves and does other things. The point is, things happen inside the cell when it's triggered from the outside. The hormone never enters the cell in this example. It's something that students commonly miss on the first exam. So I'll try to remember that analogy. It'll help you. All right. Well, let's take a break. When I take breaks in my class, I like to give about a 20-minute break. I like to empty the room so I can leave the room. I'll lock the door to make sure it's closed tight. You can leave your things in here. Let's come back in about, let's say, 1:10. 1:10, 10 out.